All right, good afternoon. My name is Hunter, and this is week 5 for Principles of Biology students. Today we're looking at cellular respiration. Now, you may notice that I'm breathing kind of hard. I've been running around a lot today, doing a lot of work that requires some energy, some heavy breathing. Now, this is topical because respiration respiration is not the same as ventilation. Ventilation is what I'm doing right now, this heavy breathing, right? This is air going in, air going out. Respiration at the cellular level is about cells creating energy, generally from glucose. Glucose is what we're going to be focusing on today. Glucose is the substrate for glycolysis and aerobic respiration, more generally speaking. Now, there are plenty of organisms that engage in both aerobic and anaerobic respiration. We're going to get into the difference between those, why it's important. Humans are, generally speaking, require oxygen for cellular respiration. That means most of our cells are aerobic in nature, and oxygen being the key feature there, the defining characteristic, this need for oxygen. So the first thing that we're going to look at here is this chart, sort of representation of not even a whole cell. We're just looking at really one compartment. We're looking at the whoop, oh my goodness, sorry about that. Ah. We're looking at the mitochondria, and the mitochondria, you may have heard, is the powerhouse of the cell, right? This should be familiar, and that is because it is the site of most of cellular respiration, right? So we have our mitochondrion as this sort of kidney bean shaped thing here in this particular representation. And around it in this sort of peach color, we have the cytosol. The cytosol, you'll remember, is the fluid inside the compartment of the cell. And the cytosol is around the mitochondrion in here. And so I guess the easiest way to go through this is from left to right, starting in this blue box with glycolysis, breaking down glucose into pyruvate. Pyruvate then gets transformed into acetyl coenzyme A, acetyl-CoA, through a process called pyruvate oxidation. That then goes into the citric acid cycle. Now, the citric acid cycle will sometimes be called, come on now, the TCA cycle, and sometimes the Krebs cycle. These all mean the same thing. Citric acid cycle. They're all the same thing. You need to know all of those names because they'll be used fairly interchangeably depending on who you're talking to, when they were educated, where they were educated. All different ways of referring to the same thing. Now, the citric acid cycle and glycolysis each produce some ATP, and ATP is that currency of the cell, you may have heard it called. It is the unit of energy that our bodies like to use the most because it has these phosphate bonds. And ATP is adenosine triphosphate. So you have an adenosine molecule bound to 3 inorganic phosphate groups, and the cleaving of that bond is what releases energy. You can see, let's see. There we go. Gotta make sure I'm not hiding important information behind my face here, which we might just get rid of at some point. So the cleaving of that bond is what releases energy that is useful within the cell. We couple this release of energy with other reactions to make them more energetically favorable. We were talking about endergonic versus exergonic reactions in the previous worksheet. This is an example of an exergonic reaction, releasing the energy stored in that phosphate bond to assist with other reactions. And this, again, I think I mentioned the balanced equation for cellular respiration is something that you will see a lot in your science education. It's a good one to have memorized. In today's context for energy, we are mostly going to be talking about ATP, but we're also going to be looking at a little bit at AMP, FAD, I think that's 2, and NADH. These are all different energy storage molecules that exist on the cellular level. So, summary for formula. The summary formula for cellular respiration is glucose. 1 molecule of glucose plus 6 equivalents of molecular oxygen is transformed to 6 molecules of CO2, carbon dioxide, 6 molecules of water, and energy. All right, so where is each of the reactants in the overall process? We have reactants on the left, products on the right. We can, maybe, yes. We can zoom out and sort of look at the overall model here. So where is glucose present as a reactant in this process? I'm having a hard time seeing my mouse, which is kind of bugging me. That's fine. We can do it with the pen tool. I'm going to go ahead and get rid of my face while I'm thinking about it. There we go. Okay. So where is glucose present? Glucose is present in glycolysis, that first step. So glycolysis, lysis of glucose. We have that 6 molecules of molecular oxygen. That is going to be present at oxidative phosphorylation, which is going to be one of our last steps here, oxidative phosphorylation. CO2 shows up a couple of places. CO2 is a byproduct of the transformation of pyruvate to acetyl-CoA. We've got CO2 here. Let's see. I want to type this so that it fits well, but I don't think I'm going to do that. Okay. In the conversion of pyruvate to acetyl coenzyme A. You might hear my dog barking in the background. Sorry about that. Okay. We also have it present in the citric acid cycle, the TCA cycle, or the Krebs cycle, all the same thing. Water shows up as a byproduct of oxidative phosphorylation again. And I'm going to go ahead and show that here. And then energy. Energy shows up a lot of places. So energy is stored in our product as ATP. It's also on the product side, or on the reactant side, as glucose, which of course shows up in glycolysis, the citric acid cycle. So the citric acid cycle in particular produces 2 GTP. The only difference between GTP and ATP is the base pair that it is bound to, adenosine versus guanine, right? So adenosine triphosphate, guanine triphosphate. And it also shows up in oxidative phosphorylation again. Glycolysis. We've got 2 cycles of glycolysis that happens. TCA cycles produces 2 GTP and an oxidative phosphorylation. And I'm just going to write a number here. We get up to 34 ATP produced for every round of oxidative phosphorylation. So that's going to be important later. All right. So in cellular respiration, the oxidation of glucose is carried out in a controlled series of reactions. At each step or reaction in the sequence, a small amount of the total energy is released. Some of this energy is lost as heat. The rest is converted to other forms that can be used by the cell to drive or fuel coupled endergonic reactions or to make ATP. The biological organism is pretty efficient, right? It doesn't like to hang on to these things. It likes to use it. Producing heat is a good adaptation if you're like a human and it's cold outside. You need to stay warm. But in general, we'd like to use that energy for other things. So, what is or are the overall functions of glycolysis? The function of glycolysis is the oxidation of glucose into 2 units of pyruvate. So glucose is a 6 carbon molecule. Pyruvate is a 3 carbon molecule. So we're getting this conversion to a smaller unit. And this would be catabolism, remember from our last worksheet. So oxidation of, I'm just going to abbreviate this, glucose, GLU, to pyruvate. This generates 2 ATP and 2 NADH per glucose. And those numbers will be important later. What is the overall function of the Krebs cycle? Krebs cycle is then taking those 3 carbon units of pyruvate, and acetyl-CoA. Pyruvate gets transformed to acetyl-CoA and carbon dioxide. So oxidation of pyruvate to acetyl coenzyme A and CO2 is a byproduct. This generates 2 GTP, guanine triphosphate, 6 NADH, and 2 FADH2 per glucose. And finally, what are the overall functions of oxidative phosphorylation? So this is the big moneymaker in terms of ATP. So the overall function is the oxidation of NADH and FADH2, or FAD, to H2O, one of our byproducts, and NAD or FAD. Although those are less important. This also generates protons, or hydrogen ions. Yes, the hydrogen is being pulled off of FAD and NAD and generates a proton gradient, a proton concentration gradient. Proton concentration gradient which is required for ATP synthase to function, which is a protein that exists in the membrane of the inner membrane of the mitochondria, which produces ATP. And I think we wrote it down, it was 34. So a lot of ATP production. Are the compounds listed here used or produced in glycolysis, the Krebs cycle, or oxidative phosphorylation? So this is just a good exercise to get in the habit of looking at these diagrams, starting to get familiar with what the inputs and outputs are at each step of cellular respiration. This is something that you are going to see over and over again, and having at least parts of it memorized will make future classes a lot easier. It will come up again whether you're in chemistry, organic chemistry, anatomy and physiology, biochemistry. It shows up everywhere. Physics, it showed up in my physics class. All right, so glucose shows up. In glycolysis, it is the substrate, so it is used. Is glucose used in the Krebs cycle? Sort of. The oxygen, let's see, we've got oxygen is used in the Krebs cycle. Oh. And oxidative phosphorylation. I guess you could say that glucose is used as a substrate for oxidative phosphorylation. Sure. Let's see. CO2 is produced in the Krebs cycle. Let's see, where else? Looking for the easy ones first. So H2O is one of our byproducts of oxidative phosphorylation, so that's a product produced. ATP, of course, is produced in all of these steps, but especially in oxidative phosphorylation. So ADP, adenosine diphosphate, plus PI, that's inorganic phosphate. So P with a subscript I is your inorganic phosphate. ADP and inorganic phosphate get used and produced at a lot of these steps. So it's produced and used in glycolysis. It's also produced, or excuse me, used in the Krebs cycle as a substrate. You get a unit of ATP out of that. And it's also used in oxidative phosphorylation in building up those ATP molecules. So NADH has that hydrogen bound to it, and that is important here. So NADH is produced during glycolysis. It is also produced in the Krebs cycle. And then that hydrogen gets broken off and NADH is consumed, is used in oxidative phosphorylation, that last step of cellular respiration. So NAD plus is the product of NADH once that proton is removed. Yes. NAD plus gets used as a substrate in each of glycolysis and the Krebs cycle, and is produced in oxidative phosphorylation. As always, if you have any questions while I'm going through this and you happen to be attending live, do drop that in the chat. I try to keep an eye on the window there as I'm going. And if you are watching this from the recording from home, feel free to watch it at double speed. I know that I try to be deliberate in how I speak, and that it takes a little bit more time, but precision of language is really important when we are talking about these really complicated processes. So. All right, the cell supply of ADP, inorganic phosphate, and NAD plus is finite. That means it is limited. Finite is not infinite. What happens to cellular respiration when all of the cell's NAD plus has been converted to NADH? So if NAD is unavailable, then the cell is unable to conduct any processes that involve the conversion of NAD plus to NADH. So looking back at our chart here, anywhere where it's used, we've got glycolysis and the Krebs cycle just in this chart, those processes would be unable to continue because a necessary substrate is missing. So both of these processes would shut down if there was no available NAD+. So if NAD+ unavailable, glycolysis and the citric acid cycle cannot proceed due to a lack of substrate. If the Krebs cycle does not require oxygen, why does cellular respiration stop after glycolysis? If the Krebs cycle does not require oxygen, why does cellular respiration stop after glycolysis when no oxygen is present? It looks like this got cropped in a weird way. Yeah, it looks like part of this question might have been cropped out, which is frustrating, but we will do our best. So if the Krebs cycle does not require oxygen, why does cellular respiration stop after glycolysis when no oxygen is present? So when no oxygen is present, oxidative phosphorylation cannot occur, and as a result, the NADH produced in glycolysis in the Krebs cycle can't be oxidized back to NAD+. When no NAD+ is available, pyruvate cannot be converted to acetyl-CoA, which is required for the Krebs cycle. So even though the Krebs cycle does not require oxygen, why does cellular respiration stop after glycolysis when no oxygen is present? So let's see, how would I write this? A lack of O2 means that oxidative phosphorylation cannot occur. Which leads to NADH rather. As a result, NADH produced in glycolysis and the Krebs cycle cannot be oxidized. To NAD+. And as we mentioned, if no NAD+, pyruvate cannot convert to acetyl coenzyme A. Which is required for the Krebs cycle, for TCA cycle. Many organisms can withstand periods of oxygen debt, or anaerobic conditions. Anaerobic, prefix an, without aerobic, oxygen. Yeast undergoing oxygen debt converts pyruvic acid to ethanol and carbon dioxide. Animals undergoing oxygen debt convert pyruvic acid to lactic acid. Pyruvic acid is fairly non-toxic in even high concentrations. Both ethanol and lactic acid are toxic in even moderate concentrations. Explain why this conversion occurs in organisms. So as we talked about in question 4, when no NAD+ is available, even glycolysis stops. No ATP will be produced. The cell or the organism will die if it cannot do some kind of respiration. So the conversion of pyruvic acid, pyruvate, to lactic acid, or ethanol, requires the input of NADH and generates NAD+. So this process is called fermentation. It's what makes a lot of foods and beverages delicious and allows the cell to continue getting at least 2 ATP per glucose. So without, let's see, in the absence of NAD+, glycolysis can't proceed, so no NAD+, no glycolysis. No ATP leads to no ATP, leads to, I'm just going to put a sad face here. Pyruvic acid, the conversion of pyruvic acid to lactate or lactic acid requires NADH and regenerates NAD+. And NAD+ is that required substrate for glycolysis to continue. And this fermentation process results in 2 ATP per glucose. So we've been doing these accounting steps, and now that accounting is going to be important. How efficient is fermentation? How efficient is cellular respiration? Remember that efficiency is about the amount of useful energy as ATP gained during the process divided by, so actually I'm going to go ahead and write this. So it's the amount of ATP gained divided by the energy of glucose. Use 686 kilocalories as the total energy available in 1 mole of glucose. So we have a new unit, 686 kcals per mole of glucose, and 8 kcals as the energy available in 1 mole of ATP. So we have 8 kcals per mole ATP. Okay, so calculating our efficiency, we take the amount of ATP produced divided by the energy in glucose. So the energy of fermentation, we said we had 8 kcals per mole of ATP. Fermentation, we said, produced 2 ATP per mole of glucose. And that is going to give us 8 times 2 is simply 16 kilocalories. Units cancel out. 16 kcals per mole of glucose. So if we take that 16, we got 2 of those. Let's see. Oop. 16 kcals per 2 moles of ATP divided by 686 kcals per mole of glucose, we should get 2.3%. So fermentation, I'm going to highlight this, fermentation has an overall efficiency of around 2.3%. By contrast, the efficiency of aerobic respiration, we've got 8 kcals again per mole of ATP. We produce 38 ATP per round per mole of glucose. Gives us a maximum of 304 kcals per mole of glucose. 304 per 38 moles of ATP divided by that 686 kcals per mole of glucose gives us an efficiency of 44.3%. Many, many, many times more efficient than fermentation. So this is why large organisms tend to be aerobic. They're more efficient at processing that cellular energy and turning it into a useful form for other activities. All right, why can't cells store large quantities of ATP? And as a hint, consider both the chemical stability of the molecule and the cell's osmotic potential. So remembering osmotic potential, if we have our cell and it's in a solution, and if there is a higher concentration of solute outside of the cell, then water will move from the area of low concentration to the area of high concentration. That's our osmotic potential, right? If, let's see, if there's a high concentration of ATP inside the cell, that means that means the cell will be hypertonic to the solution around it, and water will rush in. And you may remember that a cell in a hypotonic solution will swell, it'll take on that water, and it will burst. So that would be bad for the cell. So let's see. Actually, yeah. So a high concentration of ATP in the cell would be hypertonic to surrounding solution. And the cell could burst or lyse. So looking at the chemical stability aspect, ATP is a highly reactive at normal body temperatures. That's why it's an efficient source of energy. And it's therefore really difficult for the cell to store for any period of time. In the lab, for example, ATP is usually stored at very low temperatures, as low as -20 degrees Celsius. So ATP is highly reactive, bunch of exclamation points, at normal body temp. So it's just not very stable. It's going to break down into ADP and inorganic phosphate pretty easily. All right, given that cells can't store ATP for long periods of time, how do they store energy? The simple answer here is instead of storing ATP, the cells tend to store energy as other more complex molecules such as fats, oils, starches. So instead of ATP, cells store energy as lipids, fats, oils, same, lipids, or as starch. And starch is simply a category of storage carbohydrates. All right. What are the advantages of storing energy in these alternative forms? So ATP is a very small molecule, and that was why it affects osmotic potential, like we were talking about. It moves across the cell membrane fairly easily. These other molecules are very, very large. Lipids are very large, starches are very large, and as a result, they don't have as great an effect on osmotic potential. So large molecules do not impact osmotic potential as much. Not at all, but not as much. And they're also chemically more stable and are more stable than ATP. All right. So to make a 5 molar solution of hydrochloric acid, we had 400 milliliters of 12.5 molar hydrochloric acid to 600 milliliters of distilled water. So we already know we're going to have to do some unit conversions because we've got molar, which is moles per liter, and milliliters, which is, of course, smaller than a liter. Before adding the acid, however, we place the flask containing the distilled water into the sink because the solution can heat up so rapidly that the flask breaks. How is this reaction similar to what happens in chemiosmosis, and how is it different? All right. So in both processes, as we're adding the acid to the water, we're generating a huge difference in concentration between the two, and a proton gradient. So let's see. How do I want to? We can represent. I'm reaching back into my chemistry education to think about how they typically write this so that we can use consistent conventions here. I know that a weak acid is typically written as HA, and that H is representative of the protein, or excuse me, the proton, not protein, proton, that dissociates, and that increase in proton concentration is what makes the solution acidic. And by contrast, hydroxide ions would make it basic. So acidic and basic. So in both processes, we're going to look at our similarities. Whoa. We'll look at similarities first. In both processes, we add the acid to the water. We generate a difference in concentration between the two, or a proton gradient. We create a hydrogen ion gradient. As the proton ions flow down this gradient, that is, they mix with the water, they release energy in the form of heat. So proton flow down the gradient, that is from low concentration to high concentration, releases energy as heat. Key differences are the chemiosmosis also sets up a proton gradient. However, the energy release is controlled. As it's controlled as these protons pass through the ATP synthase molecule, and ATP is generated. So in chemiosmosis, proton flow is controlled by the protein ATP synthase, which generates ATP. Some energy is lost as heat, much of it is captured in the chemical bonds of ATP. All right. Cellular respiration, experimental scenarios. If it takes 1,000 grams of glucose to grow 10 grams of an anaerobic bacterium, how many grams, so important here, anaerobic, not requiring oxygen, how many grams of glucose would it take to grow 10 grams of the same bacterium if it was respiring aerobically? Estimate your answer. For example, if it takes X amount of glucose to grow 10 grams of anaerobic bacteria, what factor would you have to multiply or divide X by to grow 10 grams of the same bacteria aerobically? Explain how you arrived at your answer. Well, recall that aerobic respiration produces a maximum of 38 ATP max. Well, anaerobic, we had fermentation was our example. Anaerobic produces only 2. per glucose molecule. So 38 maximum per glucose molecule versus 2 maximum ATP per glucose molecule. So we could simply write that as a ratio. (38) divided by 2 would be 19. So you would need 19 times less glucose if respiring aerobically. So 1,000 grams of glucose, let's do this. 1,000 grams of glucose divided by 19 equals approximately 50 grams of glucose required via aerobic respiration. So answering this question, it means looking at the two processes comparatively, determining a ratio between them, and then using that as sort of a conversion factor. So per 1,000 grams of glucose, if that produced 10 grams of anaerobic bacterium, if it's 19 times more efficient through aerobic respiration, then we would just need 50 grams of glucose approximately. All right. Mitochondria is isolated from liver cells and can be, mitochondria isolated from liver cells can be used to study the rate of electron transport in response to a variety of chemicals. The rate of electron transport is measured as the rate of disappearance of molecular oxygen from the solution using an oxygen-sensitive electrode. How can we justify using the disappearance of oxygen from the solution as a measure of electron transport? So let's recall the balanced equation for aerobic respiration. We have glucose plus 6 molecules of oxygen becomes 6 molecules of CO2 and 6 molecules of water plus energy in the form of ATP and a few other molecules. So oxygen, the 6 molecules of oxygen is on the product side, or the reactant side. If we have an electrode that's measuring the amount of oxygen, we can justify using the disappearance of oxygen from the solution as an indirect measure of electron transport, because we know that oxygen is the final electron acceptor in the electron transport chain. So if the energy produced is 38 ATP, then for every 6 oxygen molecules consumed, or 6 moles of oxygen consumed, we would expect to see 38 molecules, or moles respectively, 38 molecules of ATP. So let's see. Yeah. For every 38 ATP, 6 equivalents of oxygen are consumed. O2 can be used as an indirect measure of energy output. C. Humans oxidize glucose in the presence of oxygen. For each mole of glucose oxidized, about 686 kilocalories of energy is released. We've seen this number before. This is true whether the mole of glucose is oxidized in human cells or burned in the air. All right, so we're given an assumption here. A calorie is the amount of energy required to raise the temperature of 1 gram of water by 1 degree Celsius. 686 kilocalories equals 686,000 calories. The average human requires about 2,000 kcals of energy per day. which is equivalent to about 3 moles of glucose per day. Given this, why don't humans spontaneously combust? We were given that example earlier where the transfer of energy was so intense that it caused the glass to explode. So as noted in question 9, during cellular respiration, the energy from the oxidation of glucose is not released all at once, right? As it's burning. Instead, each of the reactions in glycolysis, the Krebs cycle, the electron transport chain, electron transport releases a small amount of the energy stored in the molecule. So much of the energy is captured as NADH, as FAD, as ATP, as GTP. Some of that energy is lost as heat, but because of that stepwise process, the heat loss occurs at each step and not all at once. So because respiration is a complex, controlled, stepwise process, heat loss is controlled. Rather than explosive. Or all at once. So it's not just that cellular respiration is efficient, the stepwise process is important for managing the release of that energy so that we don't burn up into oblivion. All right. So D, a gene has recently been identified that encodes for a protein that increases longevity in mice. So it's increasing their lifespan. To function in increasing longevity, this gene requires a high ratio of NAD+ to NADH. Researchers have used this as evidence in support of a caloric restriction hypothesis for longevity, that a decrease in total calorie intake increases longevity. How does the requirement for a high NAD+ to NADH ratio support the caloric restriction hypothesis? So a decrease in calorie intake will decrease the rate of glycolysis in the Krebs cycle basically by limiting the reactants available for those processes to occur, right? So over a 24-hour period, there will be less NADH produced because you don't have as much NAD+ to start with by glycolysis in the Krebs cycle, and the NAD+ to NADH ratio will increase. All right, so let's write this out. So a decrease in caloric intake will decrease the rate of glycolysis and the TCA cycle, which leads to decreased NADH production by glycolysis and the TCA cycle, the Krebs cycle. Therefore, the ratio of NAD+ to NADH will also increase. E, an active college-age athlete can burn more than 3,000 kilocalories per day in exercise. If conversion of 1 mole of ATP to ADP plus inorganic phosphate releases about 7.3 kilocalories, roughly speaking, how many moles of ATP need to be produced per day for this energy need to be met? All right, so we can look at this purely mathematically, fairly simply. So we have a need of 3,000 kcals per day. We divide that by 7.3 kcals per mole of ATP. (3,000 by 7.3). This gives us 411 moles of ATP. If the molecular weight of ATP is 573, so this is, I presume this is grams per mole? Unclear. How much would the required ATP weight in kilograms? Yeah, unfortunately, I think we're missing some units in here. So if you got confused by this one, don't worry. There's a reason for that. So from our previous problem, we got that there's 411. Let's see. Moles of ATP. And we have a weight of 573 grams per mole of ATP. And we can simply run that division, or run that multiplication, canceling out our units here, gives us 235,503 grams. There are 1,000 grams per kilogram, paying attention to what the question's asking here. Simply move that 0 over, gives us 235.5 kilograms. And if you're curious, if you're from the United States and aren't used to using kilograms, the conversion from kilograms to pounds is about 2.2. So 235 kilograms is about 518 pounds. All right, explain your results. So ATP is broken down. You get ADP and inorganic phosphate, which is continuously recycled to ATP during cellular respiration. All right. So that was a review of cellular respiration. Remember that you have some extra credit that you are eligible for by turning these in by the due date on Friday. These are for completion, not correctness. So think through these. Oh gosh. Well, it was going to happen eventually. I will figure out how to get rid of these. Cool. Yeah, don't need to worry about that. Fantastic. Thank you all for your attention. Don't forget to turn in your worksheets. I will see you next week.